The overall goal of this experiment is to simultaneously detect the expression of 3 different genes in brain sections using double fluorescence in situ hybridization, followed by immunostaining.
This method can help answer key questions in the neuroscience field, such as which cell type expresses my gene of interest.
The main advantage of this technique is that you don't need a good antibody for your gene of interest to be able to localize its expression to a specific cell type.
To summarize this procedure, on the first day, fixed tissue sections are cut and hybridized with 2 differently labeled RNA probes overnight. On the second day, the sections are incubated overnight with an antibody targeting the FITC-labeled probe. This antibody is conjugated with horseradish peroxidase, which catalyzes the addition of a fluorescent tyramide on the 3rd day. Then, an alkaline phosphatase conjugated antibody is used to target the DIG-labeled probe. On day 4, this enzyme catalyzes the fluorescent precipitate formation when incubated with fast red solution. Then, the sections are incubated overnight with a primary antibody targeting a protein of interest. Finally, on day 5, this protein is visualized with a fluorophore-conjugated secondary antibody. To begin this procedure, choose a DNA clone that contains the cDNA sequence of the gene of interest in a plasmid with RNA polymerase promoters. Next, grow the DNA.